So I was asked to talk about our work as it relates to morphometrics. And yesterday in Dean Adams' talk, he talked about how his fascination with biodiversity and the wonder of morphological variation that and its functional complexities, how that fascination drove his interest in morphometrics. And I think most of us share that fascination. My entry point into the field was probably a little bit different in that I came into morphometrics because of an interest in how evolution and development were related to each other. And initially it was because I was interested in human evolution, which I'm embarrassed to say I've done virtually nothing on since, but that was my key interest. Turns out that humans are really bad experimental organisms. So studying developmental biology or evolutionary developmental biology from the perspective of human evolution is very difficult. But the research program that we have built over the years, I think, really focuses on two surprisingly complementary directions. And one of them has to do with how development structures phenotypic variation and how that in turn affects evolutionary process. And the other has to do with how development structures phenotypic variation in the context of human disease and in particularly human dysmorphology. You probably heard about this new field called precision medicine. Precision medicine is nothing more than physicians and biomedical scientists realizing that variation is important, which of course evolutionary biologists have known ever since Darwin. So I often describe what we do as developmental genetics of complex traits. So what is a complex trait? Well, here's an interesting one that we've been working on, and many others have as well. The human face. The human face, of course, is of intrinsic interest to us, but is also a highly variable, very interesting, developmentally complex trait. And human faces, like many other traits, are highly heritable. They are determined, or their shape is determined by some combination of genetic underpinnings and environmental influences. And so here are some interesting examples of faces of parents and offspring of famous people. You'll recognize some of these people, but I chose them actually because they're, well, not just because they're famous, but because they're remarkably similar, all of them. Turns out that facial shape in humans has a heritability in most populations of around 0.7. Of course, you change the environmental variance and that heritability will change. Sources of environmental variation for facial shape, for example, could be nutrition. So if you have environmental variation in nutritional status, as my colleague Paula Gonzalez has showed so well, you will change the heritability of facial shape. But by and large, it's around 0.7 or so. So it is a trait that is highly determined by genes. And yet, turns out that the genetics of facial shape is surprisingly intractable. And this is, as most of you will know, not a situation that is unique to the human face. People have been trying to make sense of complex traits of various kinds now, well, I would say ever since the finishing of the Human Genome Project. This is which created huge hopes for our ability to enumerate the genetic underpinnings of complex traits. Biomedical scientists are interested in complex traits, for example, like predisposition to diabetes, and have the hope that one could enumerate these genetic effects in order to predict individual risk scores for diabetes or to predict which drugs are going to be the best for individual people. Facial shape is no different from any of these other traits, but it is one of the more complex ones, as it turns out. We've participated in this, as it turns out, somewhat of a fool's errand in trying to find the genes that determine facial shape variation, conducting genome-wide association studies. This is the result of a study that we have done on Tanzanian children, about 7,000 Tanzanian children, a study that we did between 2009 and 2016. And we found a few significant hits that collectively explain less than 2% of the total variation in the sample. And I'm not going to tell you how much work and money went into that study, nor how much money and work went into all of the other studies that collectively, I think, explain something less than 5% of the total variation in facial shape. And the problem with all of this is not that, that individual genes aren't important in terms of determining craniofacial shape variation. It's that the way in which they do so is really complicated. So if you think about this from just a first principles basic perspective, so how does a genetic variant influence phenotypic variation or influence the phenotypic mean? So you could imagine you start with some sequence variation or a variant at a regulatory site, and that then produces some difference in either the expression of a gene, the time and place, or the three-dimensional shape of the expression of a gene, or the function of the protein that corresponds to that gene. And of course, and many complexities therein. That then in turn would produce an effect, a difference in some cellular level process, such as proliferation or apoptosis or polarity, that then in turn would affect the developing tissues in time and place that are affected by those cellular processes, which in turn would affect the morphology of the adult structure that one is interested in. So genetic variants are acting through multiple levels of process across developmental time to exert their phenotypic effects. And one of the points that I want to make through this talk, and I hope you'll come to agree with me at the end, is that morphometric approaches and morphometric theory is not just relevant to the quantification of morphological variation at this end of the scale. It's actually relevant all the way along. And the point I want to make about this is that when I first became interested in morphometrics, it was, well, I saw it as a tool. And as I used morphometrics more and more, it changed in my mind, I think, from a tool to a perspective, and then really to an approach to understanding organismal complexity and variation. So, okay. So as I said earlier, complex traits are surprisingly complicated, and the complexity that I showed here in the previous slide of variant acting through multiple levels of developmental processes, these complexities do all kinds of unexpected things. So phenomena such as a range and variation in phenotype that corresponds to a single genotype arise through this complexity. Sometimes this range is really unusual. You have situations, for example, in this model that we've been working on with Ralph Mercuccio's lab. We have a situation where within a single genotype, you get a multimodal range of variation where decision points at various places in development, and each one of these decision points can be thought of underlain by some threshold that has an underlying non-linear liability curve. That there are decision points that then end up producing this discontinuous distribution of phenotypic variation within a single genotype. So this is something that can happen. And the reason that these complexities happen is that, well, complex traits are complex. And they're complex in ways that I think we are really just scratching the surface of understanding. So if you think back to the mid-1990s and Günter Wagner's construction of the genotype-phenotype map, that's the construction that is shown here on the left. This is a way of portraying the genotype-phenotype map that captures two key concepts, epistasis and pleiotropy, the fact that individual genes have effects on multiple traits, and of course, individual traits are often influenced by the interaction of genes, so that's pleiotropy and epistasis. But in reality, the picture is much more complicated because genes are not acting directly on traits, they're acting on processes at different times and places and scales in development. So each of these processes can be influenced by multiple genes. So that would be gene interactions, but then the processes themselves will interact at different levels and times and scales in development to further complicate the interaction of the genes as they get translated to their phenotypic effects. And so understanding how these individual effects enumerate to produce variation at this level is really impossible if you haven't unpacked the stuff in the middle, if you haven't unpacked the process determinants of the variation in the phenotype. And so this is why morphometrics and the approaches therein are really central to understanding this basic problem in biology, understanding the architecture, if you will, of the genotype-phenotype map. And this is because understanding how development structures phenotypic variation requires complex morphometric approaches, requires sophisticated ways to quantify phenotypic variation. And without doing that, you have no way of investigating the complexities of the process level determinants that separate genotypic variation from phenotypic variation. So morphometrics, not just about the phenotype, it is fundamentally about disentangling the genotype-phenotype map at a very, very fundamental level. So one of the phenomena that that morphometricians, I guess, if you think of Olson and Miller as morphometricians, have developed to deal with this way in which development structures phenotypic variation is integration. Integration is not correlations among traits per se, as in Olson and Miller's construction. It's rather, or at least I would think of it as the tendency for development to produce covariation among traits. So this can be illustrated in many ways, and here's a really simple way of thinking about it. So achondroplasia is a human syndrome that is due to a mutation normally in fibroblast growth factor receptor 3. And as a consequence, people with achondroplasia have reduced growth of cartilage, and the reduction in the growth of cartilage produces the short limbs, of course, but it also produces shortening or reduction of growth of bones that depend on synchondroses, or growth plate-like structures in the skull. And so as a consequence, you have a skull that has, of course, a normally shaped brain. The mutation does nothing to affect the size of the brain, but you have a shortened basic cranium, which then produces this rather bulbous forehead, the deep-set expressive eyes of Peter Dinklage that uses the great effect as an actor, the reduction in the prognatism of the midface, and relative prognatism of the lower jaw. And so there's a set of correlated features that correspond to the effect of the mutation. Now, individuals will vary in their penetrance of this mutation. So they'll have varying degrees of reduction in the growth of cartilage. And the variation in the growth and the degree of reduction in the growth of cartilage then corresponds to the degree of extremeness of the phenotypes of these various features that are determined by this underlying perturbation. And so that set of correlations is the tendency for chondrocranial growth to drive integrated variation in the human skull. So that's integration, or at least an example of integration. Now, integration, as most of you know, is a feature of virtually every complex phenotype, or at least covariation structure is a feature of every complex phenotype, and covariation structure, of course, determined by integration. So here is a construction of the covariation structure of the human face that was produced by Jordan Bannister, a PhD student working with Niels Forkert and myself. So this is representation with 5,000 points on the face, showing that 15 dimensions explain 99% of the total variation in the shape of the face. Now, that means that if you think about the many directions in which facial shape could vary, mathematically there would be 15,000 possible dimensions, of course, because points are adjacent to each other on the mesh. It's maybe not quite that high, but it's still much higher than 15. So of the various possible directions the faces could vary, it turns out that they occupy a fairly limited portion of the total morphospace. And so that's an interesting observation. It can only happen if the variation in the shape of the face is limited in some way, limited by the processes that construct the face. And I think that is the fundamental hypothesis that we've been pursuing, and that I think lead to these to an approach that can unravel the complexities of the genotype-phenotype map. So here's another example. This is also done by Jordan Bannister. This is sexual dimorphism in the human face. In this case, Jordan actually took his own face and he morphed it to a sort of hyper female state and on this side, and a hyper male state on this side. And morphing is based simply on the average sexual dimorphism trajectory within our study sample. It turns out that if you look at syndromes that involve disruption of the underlying process of sexual differentiation, so sex chromosome related syndromes, they also produce effects that tend to fall along this same axis, this same shape axis. So there's some commonality in the developmental processes that are being affected by sex chromosome syndromes and sexual dimorphism and human facial shape overall. So, interesting observation. Now, how does this tendency for covariation or integrated variation come about in development? And I can show you many examples of this, because we've done a variety of different studies, mostly in collaboration with the Mercutio lab, with Ralph Mercutio and Nathan Young and others over the years. And I'll just quickly show you a few, because I think they illustrate some interesting properties. So this in fact was the first study of this kind that we did. This was our study of sonic hedgehog in chick embryos, and we treated chick embryos, or injected the brain of the developing chick embryos with either sonic hedgehog or 5E1 hybridoma cells that depressed repressed sonic hedgehog signaling, and we ended up then with this genotype-phenotype map, or gene expression phenotype map, that relates level of sonic hedgehog on the x-axis to morphological variation here on the y-axis. Note that this relationship is sort of interestingly nonlinear. And we also quantified some of the intermediate steps, such as midfacial cell proliferation, and here is a direct relationship between sonic hedgehog expression and midfacial shape. What I want you to notice about this is that the change in sonic hedgehog level in the fez, which is the fez is a zone of sonic hedgehog expression in the midface, produces a transformation of the face overall. It doesn't just produce a localized effect, it transforms the overall shape of the face from something that looks like midfacial clefting on one extreme to something that looks like holoprosencephaly on the other. So altering a key developmental gene produces a total transformation of the face along a continuous axis. So this is an example of how tweaking a developmental process produces an integrated set of changes in a complex morphology. And this is just a series of animations to show you what that looks like. When you think about, so one question that we asked about this later on was, is it just the level of sonic hedgehog that matters, or is it also the morphology of the sonic hedgehog expression domain? Does the morphology of the sonic hedgehog expression domain have an independently important role to play in determining the overall shape of the face? And so we asked that question a few years later, with critical assistance of Diane Hu, who was a technician in Ralph Mercutio's lab, was able to do these amazing surgeries, because what we needed to do to test this was to take chick embryos and duck embryos, because the anatomy of sonic hedgehog expression is different in these two species. And she transplanted hemi-brains, or half brains, from duck embryos into chick embryos, and saw the transformation of the face that resulted. And so this is just to show you, here's a chick embryo, here's a duck embryo. This is an example where she had transplanted a hemi brain from a chick to a chick. This is an experimental control. And here is where she had transplanted a duck hemi brain, on this side actually, not on this side, onto a chick embryo. And you get a partial transformation of the shape of the face, as well as a transformation of the shape of the fez, from the duck-like condition, from the chick-like condition to the duck-like condition, just because you've transplanted the brain, which was the original source of the sonic hedgehog signal, from one species to another. And we did morphometrics both on the fez, also on the shape of the head, and we found that the duck-chick transplants are intermediate both in fez shape and in head shape between the chicks and the ducks. So transplanting half of a brain from a chick to a duck produces a sort of halfway transformation of the chick face shape into a duck face shape. Interesting, because that shows that independently of the level of sonic hedgehog expression or the timing of sonic hedgehog expression, the 3D anatomical distribution of sonic hedgehog expression exerts an effect on the overall morphology. Also a really interesting example of how without morphometrics, one would not be able to make that insight. Here's another example. This is work done by Rebecca Green over in collaboration with Jennifer Fish, and this also is a collaboration with Ralph Mercutio, Jim Shepard, and others. And what we did here is that we used an allelic series of mouse embryos that vary in FGF8 expression. And we predicted that there would be continuous change in the morphology of the face as a consequence of that. We were also interested in phenotypic robustness, which I won't talk about very much now, but there's an interesting implication here. And we found, much like with the sonic hedgehog story, in which case we were experimentally manipulating sonic hedgehog, that when you genetically manipulate FGF8 expression, you also get this transformation of facial shape. And again, it's not localized. It's an overall transformation of the shape of the face as a consequence of varying the expression level of a single, very important developmental gene. And this graph shows this both in mouse embryos and also in mouse fetuses. Interestingly, the gene expression to phenotype map that results from this is highly nonlinear, and that has important implications for understanding phenotypic robustness, which is sort of another story. I won't go into that today. And here's my final example of this. So this is a more recent study, also done by Rebecca Green, where Rebecca and Xian Guo, the undergrad that she worked with on this, ended up relating Wnt9b expression to morphological variation in mouse embryos. And we were doing this actually because we were interested in the developmental determinants of cleft lip. And so we have a threshold model for cleft lip, and we hypothesized that Wnt9b expression was pushing ably Snell embryos. These are mice who have a mutation well, it's a mutation that affects the methylation of Wnt9b, and so there's lots of variation in Wnt9b expression within this genotype. Some embryos develop clefting and others don't, and so we predicted that the amount of methylation for the promoter site that's affected by this mutation and Wnt9b, as well as Wnt9b expression itself, would correlate with facial shape and also with clefting. And this is exactly what Becca showed, where we have a relationship between methylation level and craniofacial shape. These are the cleft embryos. These are ones that we weren't so sure about. And these are the normal embryos. And when you relate Wnt9b expression, again, to facial shape, you get this nonlinear curve. You also get a bit of a change in phenotypic variance, much as we did in the FGF8 story. But this is interesting because it shows, again, a continuous overall transformation of the shape of the face as a consequence of modulating the expression of a gene. And in this case, that continuous modulation of facial shape happens to cross a threshold, which results in a discontinuous trait, cleft lip versus not cleft lip. All right, so all of this is to make a single point, and that is that perturbations to developmental processes have integrated and often highly pleiotropic effects. And this is because genetic variants are acting through these multiple levels of developmental process as they exert their effects on the phenotype. And this is, of course, a central hypothesis in evolutionary developmental biology, but it has implications that I think are poorly understood in terms of what they mean for the quantitative genetics and the developmental genetics of complex traits in general. So this not only implies that variation is highly structured by development, but it also implies something much more fundamental, which is that the dimensionality of phenotypic variation may be limited to the directions of variation that correspond to how those developmental processes can affect the phenotype. And if that's the case, that's fundamental. And it's fundamental because it's a way to simplify this enormous complexity of how highly polygenic traits like stature or facial shape that may be determined by thousands of different variants are converging to something that would constitute an explanation for phenotypic variation at the developmental genetic level. So take stature as an example. Recent studies of stature have shown basically an asymptotic relationship between the number of potential determining loci and phenotypic variation. How many genetic variants in the population determine stature? Well, this depends on how many individuals are in your GWAS. The more individuals you have, the more loci you get. The largest number I've seen is 22,000. It's probably arbitrary. If you genotyped everybody in the world, who knows how many you would get. But there's this really long tail of small effects whereby virtually anything genetically is going to have some effect on stature if you have a large enough sample to measure it. Facial shape variation is probably similar. So what that means is that genetic explanations that enumerate individual gene effects are not particularly meaningful. They don't tell us anything other than that the trait is heritable. And so we need a different way to approach explanations of these traits. And the fact that the dimensionality of phenotypic variation, or at least the hypothesis that dimensionality of phenotypic variation may be limited by development, offers the hope of generating such explanations. So what I'm going to do in the remainder of this talk is to derive 3 predictions from this central hypothesis and show you the evidence that we have or that are confirmatory for these hypotheses. At least don't reject them. And also for these predictions, and hopefully convince you that this is a productive way of looking at the developmental genetics of complex traits going forward. So the first of these predictions is that genetic effects converge on a limited set of directions of phenotypic variation. The second prediction is that phenotypic effects of genetic variants converge on process level determinants of phenotypic variation. In other words, the genetic variants aren't just a random assemblage of things. There's some meaning in which genes are involved in which direction of variation and which trait. And the third is that mutations of major effect recapitulate the background covariance structure. And this is maybe somewhat obscure, but it's a consequence of the limited dimensionality. If there are only so many directions in which you can vary, then you don't need that many genetic perturbations to capture those directions. So genetic variation caused by the 10 million or so segregating polymorphisms in the human genome may not be that different from, say, a set of 30 or 50 different mutations of major effect because, well, there aren't so many directions to vary. Does that make sense? Actually, nobody can answer me, so I'll just keep going. So let's examine these predictions. This is from a study from a few years ago that Michaela Pavichev and Philip Mitterocker and I did, where we examined the directions of deviations from the in the crosses of the 8 founder strains of the of the collaborative cross panel. So the collaborative cross is a big mouse breeding project that is produced by crossing together 8 different strains of mice. And so what we did here is we took all 64 different crosses of these 8 founder strains, and we examined the phenotypic directions of these crosses. And we found that there is a common pattern of dominance, and that there is also a common tendency for the crosses of the founder strains to deviate in particular directions. They don't just go often in the random direction. They have a non-random tendency to deviate in the direction away from the mid-parental mean. And this is a result that you could only get if there was an underlying tendency for variation to be structured by development. So to examine this further, we've been working on, well, mainly on the diversity outcross project, which is also derived from the collaborative cross. And so this is a large recombinant inbred strain project in which we have taken, or we have, but the collaborative cross consortium has taken these 8 founder strains and crossed them repeatedly over multiple generations. I think it's up to it's in the high 20s or low 30s right now. And then you create this panel drawn from different generations, in which you can examine the effect of variants derived from each of these 8 founder strains on phenotypic variation. So sort of an 8-way cross to generate phenotypic variation. And I should say this has been a somewhat controversial project, and it's controversial, I guess, because of the range of variation among the founder strains. 2 of the founder strains are actually subspecies of mice. They're different subspecies, and so that has produced some difficulties in the breeding. But it certainly means that there's no shortage of genetic variation in this panel. So we started by just doing a straightforward QTL, and this is work by David Cates, postdoc in the lab, where he examined or studied QTLs for the main principal components for craniofacial shape, and went beyond this to examine the extent to which the hits for these QTLs were genes that were more or less likely to be involved in craniofacial and bone development. And so we did find, indeed, that in the first principal components, the components that explain the majority of the variation in the shape of the face, that there is a non-random tendency for the QTL that come up to be in genes that are associated with growth, skeletal system development, sensory organ development, cartilage development, and bone development. So that's good. It means that there's some structure to this. It's not just a bunch of random stuff, even though craniofacial shape is extraordinarily polygenic. And so building on this foundation, what we've been doing, and this is work done by David Aponte, PhD student in the lab. What we've been doing is using an approach that was developed initially by Philip Mitteroker to identify latent or underlying variables in genomic data and relate that to central tendencies of covariation in the morphological data. Now, this is an interesting approach for a variety of reasons. I think it's transformative in complex trait genetics, but it's obviously it's obviously an approach that is highly influenced by morphometrics. I think the initial idea that spurred Philip to take this was basically, how would you do a PLS for genetic data on one hand and phenotypic data on the other? So this is sort of thinking about this problem as a morphometrician would. And when you do that, you come to the realization that you can take these 2 data sets and draw from them underlying latent variables that have some correspondence to, for which you can test correspondence for underlying process associations using gene annotation data, and see the extent to which these patterns in the genomic data actually drive phenotypic variation in a trait like the skull. And so this is the basic idea that David took and developed what we are calling in the lab the MGP approach to, or the multivariate genotype-phenotype approach. So, and there's an app that corresponds to this, and I'll show you the link to that a little bit later. You can go and play with it if you're interested. But one thing we can do with this is that we can take the genes that have some gene annotation association, like chondrocyte differentiation or FGF pathway, and then see the extent to which the segregating variants in the DO mouse panel that are associated with those genes produce phenotypic variation. And so here's 2 examples: chondrocyte differentiation on the one hand, and the FGF pathway on the other, and here are individual genes that have high loadings on the phenotypic effects that are associated with this underlying latent variable. And another thing that David did is that he then took mutations or mutant phenotypes from our database of mouse mutants that involved mutations in those same pathways or processes, and he compared the directions of effect. So for example, here, he compared the direction of effect for bone morphogenic protein receptor 1B to the chondrocyte differentiation pathway. And for FGF8, here's FGF10 compared to the overall FGF pathway, and found that in many of these cases, these are highly correlated. When you look at these processes overall, so they have a non-random association with phenotypic variation overall. So this is an interesting plot here, which shows the first 4 principal components of craniofacial shape and their association with randomly chosen process effects based on Go terms that had been run through the MGP analysis. And it shows that for the first principal component, there aren't that many processes that are driving that variation, but the correlation is very high. In other words, those processes are strongly determining craniofacial variation for along the first principal component. And then as you go down the list of principal components, you get to larger numbers of processes but smaller effects. In other words, a more disintegrated pattern of variation. And these processes themselves are also correlated in terms of their phenotypic effects. So, and this is also what one would expect given the model that I showed you earlier, where you think about the genotype-phenotype map as processes that are interacting and producing integrated phenotypic variation. The phenotypic effects of different processes, as determined by the MGP method, are also co-vary with each other. So cell proliferation co-varies with, in this case, fibroblast growth factor receptor signaling pathway, etc. So there are multiple levels of pattern to dissect here. So that deals, I think, with the second prediction. I could show you other data, but this hopefully is enough to convince you of the idea that the genetic effects tend to converge on processes that drive variation. That you don't have that there is structure in the way in which individual genetic effects relate to each other to affect processes and then in turn affect the phenotype. So the third prediction, and the most obscure one, but also the most easily testable one, is that mutations of major effect recapitulate the background variance covariance structure. Sorry, the background covariance structure, not the variance structure. And so I'll show you two lines of evidence for this. We'll start again with the MGP data from David Aponte. So here we have the MGP phenotypes projected onto a principal components analysis of the diversity outcross mice, along with a sample of 30 different loss mutant genotypes. And it shows that there is a tendency for mutations to follow along similar directions as the processes that those mutations are thought to affect. And if you just test this quantitatively, the yellow squares are ones with higher correlations, and we see that that there are for some of these processes fairly high correlations with mutations that affect those processes. So the basic picture is that many, but by no means not all, mutations of major effect occur in directions that are consistent with the effects of major processes affected by those mutations. So that's one way of testing this prediction. But let's look at another way. So another long-term and large project that we've been working with in our lab, and this is a large collaboration involving principally Ophir Klein, Rich Spritz, Francois Bernier, and Niels Forkert and their groups. We've been analyzing and recruiting patients with human syndromes, with genetic syndromes, and doing 3D facial shape imaging and analysis of these faces. And we've built up over time now a database of about 5,700 subjects with known syndromes. Total database is over 7,000 subjects right now. And we have, in some cases, the relatives of these patients, and we also have some unaffected controls, but we've been using also other sources of unaffected controls, such as the human GWAS facial shape data that we've worked with previously. So this list shows sample size distributions for various syndromes, and age distribution, etc. And what the point I want to make about these data here is that one can treat the syndromic means, in this case, as individual genotypes. So this is a principal components analysis of the mean facial shape effects of different human syndromes. And so this is, I think, for 64 human syndromes. And when you, and this just shows you also some heat maps showing what these, what the syndrome-associated variation looks like, and also some heat map views of particular syndromes that happen to fall on the extremes of these PCs, showing that they are somewhat similar, not identical, but somewhat similar. So this is suggestive. When you test this quantitatively, though, it turns out that the covariance structure for the 64 syndromic face means is virtually identical to the covariance structure of the background population from which they are drawn. And it's virtually identical also to the covariance structure of the of unaffected individuals. So in this case, it was the comparison was to the Caucasian GWAS study of Weinberg et al., about 3,000 individuals. And this will be the correlation, or in this case, the random skewers correlation is 0.9 to the control sample, and the covariance structure for the syndromic means is sorry, the covariance structure for the syndromic means, the syndromic sample, and the background unaffected sample are virtually identical. In other words, and this is the remarkable observation here, 64 syndromes virtually capture the variance-covariance structure of the human face. Now, they don't capture the variance, though, because in fact, many of these syndromic effects have elevated variance. In fact, if you look at the relationship between the variance within syndromes and the shape distance to controls, there is a tendency for more severe syndromes to be more variable. There's also a tendency for syndromes that are more variable to have higher levels of integratedness or higher scale variance of eigenvalues. And this is simply because of effects like the achondroplasia story that I told you earlier. If you have variable penetrance in some process that is producing a set of correlated effects, and the more extreme the perturbation to that process is, the more variance you will have in penetrance, and the more that variable penetrance will drive integrated effects. So integration tends to increase in this case with the magnitude of the perturbation, which is, it's a common observation, but it's a frequently misunderstood one, because people often think of loss of integration as being a bad thing. In this case, or somehow same thing as a decanalizing influence, which it is not. It's simply a measure of the extent to which some underlying developmental process is driving a set of correlated effects. And in the case of many syndromes, which are undeniably bad, the phenotypes can be highly integrated, can have a very strong covariance, because the underlying perturbation is producing a set of correlated effects, and the amount of variation in that perturbation among individuals is driving an increase in the strength of covariation or integratedness. So here's another way of approaching the same problem with these data. So this is an interesting study that's being done by Hannah Hoskins, who's a PhD student in Peter Clay's lab, and Peter Clay's is collaborating with us on this. And the question that Hannah is asking is, can you think of syndromic phenotypes as traits? So rather than the syndromic phenotypes as determined by the major mutation that is normally associated with those syndromic phenotype, with that syndromic phenotype, what if you think about, say, the achondroplasia-like face as a quantitative trait, regardless of the FGF3, the FGF3R mutation. So if you, and there's one example, this has been done now for several different symptoms, but if you project the achondroplasia phenotype on our Tanzanian facial shape data, and none of those individuals have achondroplasia, by the way, you get an interesting result. You get a whole set of hits in the GWAS that are associated with variation in this achondroplasia-like phenotype. And here's the strongest one that she found, this chromosome 4, FAM53A, which is, we have no idea what that gene does in craniofacial development, but it has been associated with clefts of the soft palate. And there is, interestingly, also an association with the FGFR3 region, but not as strong, but many other associations. So this means that treating syndrome-like effects as phenotypes reveals a polygenic basis for the shape effects of those phenotypes, which again could only happen if the syndromic effects represent directions of variation that are being determined by underlying development processes, but those mutations happen to push individuals to an extreme along, but normal variation is also acting on. So syndromic phenotypes represent extremes of normal variation is what that basically shows. And I'll come back to the previous slide that sort of shows that. These are the syndromic means here. And if you look at these distributions along individual PCs, you'll often see that they'll have long tails with bumps, and those bumps can be individual syndromes that produce effects that are extreme along those directions. So, and I forgot to mention this, that all of these data here are projections on the variance covariance structure. The plots here are projections of the variance covariance structure for the syndromic data onto the background population, the Caucasian GWAS data. And so the Caucasian GWAS data varies in exactly the same directions as the syndromic means, but the syndromic means have extreme extensions where particular syndromes are pushing along those same axes of covariation, but to extreme degrees. So syndromes are extremes of normal. All right. Just as an aside, if you want to play with any of this, David Aponte has been putting together a set of apps in which you can explore data and do both MGP analyses and also look at syndromic variation. And that link is shown here. I can certainly ask Ugo to send that around if people are interested. But you can find it here at genopheno.ucalgary.ca. All right, so what are the implications of this then for complex traits? So I think what I'm hoping is by now an obvious conclusion is that for highly polygenic traits like facial shape and by extension other traits that people are interested in like stature, maybe things like diabetes, pursuing developmental process of determinants of variation is much more likely to be fruitful than enumerating individual gene effects, particularly if those individual gene effects are a very large number of genes. Those are unlikely to provide fundamental insights, but the processes that those genes act on are likely to provide fruitful insights into the underlying determinants of variation in those traits. This also means that development and morphometrics, or the quantification of the results of development or developmental processes, needs to be incorporated into genetic hypotheses rather than serving only to validate after the fact. To do all this, though, we need new approaches. In other words, and in particular, developmental biology needs more morphometrics. And I'm going to just tell you about two directions in which I think we really need to expand the way in which developmental biologists do business in order for this vision of the developmental genetics of morphology to be realized. One is that we will require better methods to integrate across levels of the genotype-phenotype map. In other words, we need to be able to integrate, particularly within individual embryos or specimens, but there are other ways to do that. We need to be able to integrate variation and gene expression to cellular processes, to tissue level processes, to phenotypic variation. And the approaches and methods of morphometrics are key to doing that. We have been working in this area, and I hope that others are doing so as well. And so one thing that I'm particularly excited about is the growing ability to quantify cellular processes in 3 dimensions, and at the same time do morphometric analyses of those individual specimens and extract meaningful patterns from the spatio-temporal variation in cellular processes and relate them to morphology. That's a major direction that we're pursuing in the lab, and this just shows you some images that come out of this work. We've also, and particularly Rebecca Green, has been working on ways to quantify gene expression and relate that to morphology, and we've had papers published on that in the last couple of years. And we've been increasingly integrating morphometric methods from the brain imaging world to try to bring these messy and difficult to work with data sets together in ways that make sense. And so, Lucas Loversio, sorry, his name is misspelled here again. Lucas, if you're listening to this, I apologize. Is a key person in the lab working in this area, as well as Rebecca Green, Sian, Marta Marcini, and Andreas Dauder. So the other direction that is also required for this vision to be realized is we have to move away from highly individualized, manually intensive data collection, because the kinds of approaches that I'm talking about require often large samples, requires the ability to standardize across studies. So, for example, the ability to relate variation across multiple mutant phenotypes. That requires that we are collecting exactly the same kinds of data every time we analyze a mouse mutant, for example. And that's really, really difficult to do if you're relying exclusively on manual landmarking. And so, through the work of people like Jay Devine and Chris Percival, who was in the lab and is now at Stony Brook, and Marta Vidal, who we just heard at this conference, is also working in this area with us, and Jordan Bannister. We are working on ways to create automated methods for phenotyping that are both efficient, standardized, but also flexible. And by flexible, I mean the ability to take the same underlying data sets, such as volumetric registrations, and extract from those data sets quantifications that are individualized for different hypotheses. So these are just, here's an overview of our mouse embryo phenotyping pipeline. And this is just a particular set of landmarks that Marta had created to study brain-face integration. But the key point here is that it's not the individual landmarks that are important, but the registrations of these images onto each other that then allows Marta, in this case, to extract whatever landmarks she wants to test their hypotheses. And those are the kinds of approaches that will be necessary for us to be able to integrate across large data sets and build a more comprehensive understanding of the developmental genetics of complex traits. Now, automated phenotyping, and particularly automated landmarking, is difficult. And for those of you who have worked in this area, you would know this. There are a variety of artifacts and problems that one needs to be careful with. And for nonlinear registration of volumetric data, the most important of which is the tendency for extreme phenotypes to get pulled back in towards the mean. In other words, the further an individual is away from the atlas, the less accurate your quantification of that individual can become in a way that's not so simple. It's not so it's not so easy to correct for. So Chris Percival has done a great analysis of this effect. Jay Devine has a machine learning enabled approach that uses manual landmark data to correct for this effect. It works really well, but it does have the disadvantage that then you are confined to using automated versions of the same manual landmark data sets, or at least you would need large manual landmark data sets to train the machine learning algorithm to better position the landmarks. And so there's a bit of a trade-off there. But the bottom line is that these methods are important. They are still in development and do require careful validation, but I think they are absolutely necessary for us to take morphometric analysis in the context of developmental genetics to the next level. So just to conclude, what I hope I have convinced you of is that morphometrics is about more than quantifying external structures or adult phenotypes, or even ontogenic series of bones. I mean, those are all useful things to do, but morphometric analysis can be applied in a wide variety of settings in realms that would previously that are normally thought of as belonging to developmental biology. In fact, I think integrating morphometrics into developmental biology is a key conceptual leap that needs to happen. And for the reasons that I've been convincing you of today, but also for the reason that without doing that, it's impossible to bring together the understandings of developmental genetics that come out of genetics on the one hand and development on the other. And the reason for that is sort of stupefyingly simple if you think about it, and it's the fact that genetics is about the study of differences. So quantitative genetics is about variants that contribute to phenotypic variation. The concept of variation is built, it's a core concept within genetics. But developmental biology is about gene function and mechanisms. These two fields don't talk to each other unless you ask the question, how do genetic variants act on mechanisms to produce differences among individuals? That question doesn't exist in either field. It only exists when you bring morphometrics into the equation. And to do that, we need to develop better and more sophisticated tools for quantifying phenotypic variation at all levels of the genotype-phenotype map. And that is a major goal of our group, and I hope of others as well. And with that, I would like to conclude and thank the people in my lab and our collaborators, without whom none of this work would happen. I apologize to everyone in the lab for any mistakes that I've made in the characterization of your work, or if I've forgotten to mention anybody. And I also thank our funding sources. And with that, I'm happy to take questions.
Thank you very much, Benedikt, for your wonderful, I have no words for you. It was very, very, very impressive. So now we have the round of questions. The first question is Miguel Contreras. Please, Miguel.
Thank you. Thank you, Doctor, for your wonderful presentation. I was wondering if you have thought about the role of admixture and how the process of gene flow between humans in the last 10,000 or, well, since we originated as a species, what is the role of admixture in modeling the variance covariance structure, and what are the implications of that? The main databases that we have right now are like European biased, and how that fact would be introducing some sort of error into our predictions about variability in the facial.
Yeah, wow, that's a huge question and an excellent question. So where to start? Have I thought about this problem? Yes. Smarter people than I have thought about it as well. The question of the extent to which the clear European bias affects our understanding of human genetics and genomics is probably the most important one. And I think there is a clear bias there. The most important part of the world to sample better is Africa, because it also has the largest amount of genetic diversity. Now, one thing to keep in mind, though, is that human genetic variation is much less geographically structured than, well, than many people assume. So something like 85% of all of the segregating polymorphisms in the human genome are found in all populations. And something like 7% or so of these variants group along major geographic lines, and the remaining 7 to 8% vary along smaller geographic lines. And so humans are less geographically structured than one might think. So, for example, the genetic basis for grouping people into races quickly falls apart when one looks closely at the data. We've done analysis of the facial shape variation in Tanzanian children compared to European-derived North American children, which is in a paper by Jacinda Larson, did her PhD in our lab 4 years ago. And we showed there that that the difference between those two groups only represent about 5% of the variation in facial shape. And that's an observation I find interesting on multiple levels. First of all, it's a bit less than you would expect given the genetic facts that I told you. You would expect maybe something like 15% of the shape variance would be would be attributable to the difference between those groups, but it's about 5%. And the other thing that's interesting is that most people working with those two groups of children would have no trouble identifying them, even on black and white photographs. So what that tells me is that human faces are much more diverse within populations than we think, because we are really good at picking out differences among people. And we have a very sophisticated machine learning algorithm in our brains that extracts features of interest, or at least extracts features that we can use to impose typology where really there isn't an underlying typology to impose. Most variation in human faces you find within every population. That doesn't answer all your questions, but maybe it gives you a bit of a perspective on that. And I don't want my remarks to be interpreted as it not being important to sample across the globe to get around the kinds of biases that we are seeing when you have very European-focused data, and that's because it can easily happen that disease-causing variants that may be important to find are not important in European populations, and they may be occurring elsewhere, and also because European populations are sampling less of the diversity in the human genome than, say, African populations are.
Wonderful, Benedikt. So we have another question now. There's Daniela Flores. So let me. Okay.
Hi, hello. Can you listen to me?
Yeah, I can hear you.
Okay, good. First of all, thank you so much for your talk. It was amazing, and thank you. Thanks for all organizers of this Congress. About your talk, I think that my question is very general. I think that considering that during development there is an integration of different levels, and as you said, the genetical level and then the morphological level, but in the morphological level it is possible, I don't know if it's possible to integrate somehow the development of skeletal structures and with the soft tissue structures, because in some stages, generally in post-morphogenetical stages, it is possible that this soft tissue can act as a constraint of development and also as a promoters of this skeletal structure. So I don't know if you can give me some orientation about this.
It's a great question. It's a great question. And so the last question was about is our view of human genetics European biased? This question is about is our view of organismal diversity bone biased? And I think that both of those are true. There are absolutely, so the reason that people measure bone so much is that they're easy to measure and you find them in museums. But it's very clear, and again depending on the question, but it's very clear that there are in the majority of analyses that have been done on bones, including the ones that we've done, you would have gotten much more interesting answers if one had imaged soft tissues as well. If you're doing, say, volumetric superimpositions of MRI scans of whole individuals or CT scans of whole individuals with soft tissue contrast and quantifying morphology, not this of the skeletal structures, but of the surrounding soft tissue structures, we might have a different view of organismal diversity if we had been proceeding in that way. But that's limited by technology. In science, we see what we are able to see. And we are bone biased because that is the easiest source of data, but you're absolutely right.
Yes. Okay, thank you so much.
Thank you very much, Daniela. And I think we don't have more questions.
Well, thank you very much. And I thank everybody for listening. I think this has been a fantastic group. And I really applaud you for doing this.
Was a fantastic talk. So thank you very much, Benedikt, for your time and to be our second keynote talk.